All right, so 4 o'clock. Today we're going to cover topic 8. We might break into topic 9 a little bit too. Topic 8 is kind of a short one, and topic 8 and 9 are kind of related in terms of one's around energy and energetics, the other's around enzymes, and so they're same part of the textbook too. All right, so topic 8 is cellular order and energetics. So make sure you have a piece of paper today. I'm going to do another diagram and a couple other notes as we go along. And so topic 8 and 9 are kind of short units, and they're units to kind of just get us ready to talk about respiration and photosynthesis. So just reviewing a few things around energy and ATP and all those sorts of things. So like I said, they're relatively short, and then hopefully they'll help us to be able to comprehend respiration and photosynthesis in a lot better detail. Before I get into topic 8 here, what I want to do is actually play this video for you. This is one of these fancy animations. This was actually done in collaboration with Harvard in the States, and they spent a bundle of money on it. And it's called The Inner Life of a Cell. And there's a whole bunch of things going on in this video. It's actually really cool. If you really understand a lot about cell biology, you're going to see a number of things in there. And you're going to be able to see a number of things that we've talked about as well. And if you're interested in all the details on this, you can find information about it online. But I'm going to play this for you, and I'll try to point out a few things as we go along. So here goes, the inner life of a cell. By the way, they did their best to make everything as accurate as possible in terms of protein structure and those kind of things. So here we go. So what you're looking at, by the way, are some white blood cells moving around. That's what you were seeing. And there's some phospholipids and some proteins moving around in the phospholipid bilayer. So we're going inside the cell, we're going to see some cytoskeleton. So those are actin fibers or microfilaments. So those are just inside the membrane. You can see they're assembling and disassembling and helping the cell to move around. So which one is that? Those are the microtubules. And there we go. There's a motor protein carrying a vesicle. I told you they were kind of like little feet. They move around. And the ATP is hydrolyzing during that process. Upper right, we have a mitochondria. Bottom left, we have a centrosome. And there are nuclear pores, and that is RNA coming out of nuclear pores. And it's going to find the rough endoplasmic reticulum. That was a ribosome. Some mitochondria. Or sorry, that's not the mitochondria, that's the rough endoplasmic reticulum. There's the Golgi. So what they're trying to show you in this video basically is the production and secretion of some sort of protein that's getting secreted from the cell. And there goes our little white blood cell moving along. And you can see in the end that white blood cell is getting called to an infection. It's crawling in between some tissues to respond to some sort of infection. Okay, so I hope you enjoyed that video. the reason I wanted to show it to you is just to kind of show you some of the things that we talked about in class already. And the other reason I want to show you is that unfortunately, you know, when we're talking about cells in class and when you're reading about them in textbooks, often you're just seeing pictures. And the reality is cells are very dynamic. There are things happening all the time. There are organelles moving. There are things being assembled and disassembled, and they're very, very dynamic things. And of course, all of these things require energy, which is what we're talking about today. So some definitions for you. One definition is metabolism. I know we throw that word around a lot. People talking about trying to lose weight, and they're talking about maybe boosting their metabolism or something like that. But metabolism really just means the set of chemical reactions in your body or in a cell or something like that. So when people are talking about boosting the metabolism, they're really just talking about trying to you know, maybe have a little bit more chemistry or more reactions happening in order to, I guess the idea is to burn fat and lose weight kind of thing. So some ways we can further classify these reactions is we can call them either catabolic over here or anabolic. So I want to talk about what these things mean here in terms of what's going on with these reactions. So catabolic reactions, these are breaking down type of reactions. And the example you can see I have here is the catabolism of glucose. So we're breaking glucose down. It has 6 carbons, and we're breaking it down into carbon dioxide and water. Probably some people who are eagle-eyed are saying, hey, that chemical reaction is not balanced. And you know what? That's okay. I don't really like balancing equations, but it is breaking down glucose into carbon dioxide and water, which are more simple compounds. Notice the other part of catabolic reactions is usually they're releasing some sort of energy. Anabolic, on the other hand, is the opposite of that. So anabolic is where we're building something up. So we're going to require the input of energy. So usually we're taking something more simple, like amino acids, and we're building something more complex, like a protein, which is a string of amino acids. The way I usually remember which is which anabolic and catabolic is I think about, you know, you might hear about these football players or people in the gym taking anabolic steroids. So why are they taking anabolic steroids? Because they're trying to build up their muscle mass. So anabolic is building, catabolic is breaking. So let's talk about energy as well. We have lots of energy apparently in Fort McMurray in the oil sands, so people like to talk about it a lot. And if you think about energy, it has many forms. For biology, the most useful type of form of energy that we like to talk about is potential energy. And what do we mean by that? I mean energy stored usually in chemical bonds. So if you take a look, there's a kind of a mini reaction there. You can see how 2 things get together, and it takes energy to put them together. So now that they have a new chemical bond, when that chemical bond is broken, it can release energy. And energy, of course, is by definition, it's something that can do work. And we'll talk about what some of that work is, but work could be moving your cell around. It could be building new proteins. So here's another way to think of potential energy, right? Is you have, it's stored in a system. So in this case here, we've got Katniss, and she's pulling her bowstring back, and energy is stored in that system. And as soon as she lets go, the energy will be released. It'll do the work, which is moving the arrow. So here's a little diagram from the textbook, by the way, talking about energy. And you can see that we have some different divers. And for example, we have a diver in the water and a diver who's high up on the platform. And so the diver in the water has low potential energy, and the diver high on the platform has stored potential energy. How did he or she get there? Basically by climbing the ladders. And so climbing the ladders, that's kinetic energy because it's movement. And eventually the energy gets stored as potential energy in the form of being higher up in terms of stored as potential energy due to gravity. That potential energy can be converted back to kinetic energy once he or she jumps off. So this has a lot to do with something that we call thermodynamics. And you probably have heard about thermodynamics a little bit through chemistry courses, or if you've taken some physics courses. And it can get really, really complex. And if you are in Chemistry 102 this semester, you're going to be talking a little bit about thermodynamics. So I want to talk about a little bit in this class and kind of relate it back to cells. There's a few laws of thermodynamics. The most important ones are probably number 1 and 2. We're going to talk about that. The first law of thermodynamics is that energy in the universe is a constant thing, meaning we can't create energy, but we can convert it between things. So if you take a look here, we have this bear here is not creating any energy, but he's consuming it from a fish. So the fish, of course, has energy stored in its chemical bonds, and the bear is going to take that energy and probably convert it into new chemical bonds, or maybe convert it into some sort of movement. So I'll come back to this in a moment and relate it to cells, so don't worry, I'll come back to that. Second law of thermodynamics. Uh-oh, what happened to my video? Let me just try that again. The video is doing something weird. Maybe when I press play, it'll do something normal. Hold on. Oh, there we go. That's better. So let me play this video for you while I'm talking. There's a bunch of these on the internet, by the way. They're super cool. This is one of these time-lapse photography. So they just have a camera on the strawberries over like a 2-day period, probably longer than that. And you can see what's happening is that the strawberries are, well, they're dead and they're dying, and they are basically degrading and breaking down. And the mold is obviously speeding up that process. And this relates to the second law of thermodynamics. And the second law has to do with something called entropy. And don't want to get caught up in talking about entropy and all that. This is not a chemistry class. But entropy basically means disorder. So what it's saying is that energy transformations result in increasing disorder. And what does that mean? It means that, like, if you have anything in life, whether it's a car or a bedroom or a fridge, it means that without effort, these things just get messy. So what does that mean for the cell? It means for a cell, if you want to stay alive, you want to stay organized, it's going to take energy to do so. So like I said, I'll sum these up here in a second in terms of how we can relate them to cells. So obviously that's one of the huge one of those big characteristics of life is that life is organized. We see order in these things. And like I said, there's a cost to that, and that it takes a lot of energy. So here's our little cell, and you can see in terms of relating this to order, the cell is increasing in order, and the cost is to its surroundings. And surroundings means food and whatever else it's metabolizing. So let's sum these up. There's the first law, conservation of energy. Second law has to do with order and disorder. So like I said, I wanted to make a note on this in terms of, I'm calling this the laws of thermodynamics for cells. So the first law of thermodynamics is that cells cannot destroy energy. Sorry, cannot make or destroy energy. Make or destroy energy. But, there's a big but here, but they can convert between different forms. Different forms. So for example, if you're a plant, maybe you're converting from light energy to chemical energy. So let's say that example, from light energy, chemical energy. And by chemical energy, of course, I mean chemical potential energy in bonds. Okay, second law of thermodynamics. Second law of thermodynamics basically says it takes energy to stay alive. It takes energy to get organized and to stay alive. Organized and stay alive. I feel like there was something else I was going to say about that. Let me just double check my notes here. Yeah, I guess just sort of kind of on the same note is that basically that means that cells must continually consume energy, right? Cells must continually consume energy to keep themselves organized. And alive. So I guess that's just a rewording of my first sentence. So that is kind of the important thing to know about thermodynamics, unless of course you are in Chem 101, where you are going to talk about a few more things. I do have a few other things to say about thermodynamics and a few other kind of definitions that are important to know. And if you continue on in cell biology and biochemistry and things like that, you're going to see a lot more of this stuff. All right, so hopefully you got all that. So another thing that has something to do with thermodynamics is this term here, free energy. So you can see free energy is represented by this G here. The G, it's actually named after a guy named Gibbs. And so sometimes it's called Gibbs free energy. He was a chemist who a long time ago studied this kind of system. And so we're actually looking at the change in free energy. And you can see from my definition here, it says it's a measure of a system's instability. Another way to think about it is free energy is kind of, if you think about all the energy available in a chemical transformation, some of that energy is lost as heat and it's not useful. This is kind of the useful part of that energy, right? So there are different ways to break down what delta G means. And I am not going to come back to these formulas ever again in this class, but if you do take Chem 101, you probably do a little bit more of this. You can see kind of how this breaks down, right? Here's my delta G down here. Like I said, the total energy, and some of it is lost as heat and disorder, and that's not the useful stuff. So the delta G is the useful stuff. So often when we talk about chemical reactions, we're looking at kind of that amount of energy that's released or gained in a system that's useful. And so it's a good gauge to kind of understand what's going on in a reaction. So let's kind of just talk about what this means a bit more. I know it's not something, it's a little abstract and takes a little bit of time to think about a little bit. So take a look at this. I have 3 systems, and all 3 systems in the top part are going from a higher energy state, and in the bottom part they're going to a lower energy state. So the first system here, you can see we have a diver. And we talked about that already. The diver is in high energy state because he climbed up the ladders. And when he jumps into the water, he's going to lose that energy. It's getting converted into kinetic energy. And then when he gets down to the bottom, it's at a lower state. So that system there is losing energy. It's going from higher energy to lower energy. And specifically, we're talking about the G, the delta G change, and which is the useful energy. System 2, you can see some diffusion. So it's just some molecules put in water and they dissolve and spread apart. So they're going from organized to less organized, more energy to less. The last example is a chemical reaction. It's breaking down glucose and breaking down glucose into carbon dioxide and water. And so it's going from a complex, more organized molecule to something that's a bit more disordered. So this is what happens when a system loses energy. We say that the delta G is negative. Okay, so this is something important to remember. So I'm going to write a note on that, in fact, because it's important enough to remember. So let me just pull my laptop a little bit closer. So there's 2 notes to think about here. So first is if delta G is negative, so what does that mean? Okay, this means the system usually meaning the chemical reaction. So the system is releasing energy. Or you could think of it as losing energy if you like as well. Losing energy. So this, I will say, is usually catabolic. So if you think about the breaking down of glucose, we're releasing energy, usually catabolic. What else is going to say about this? And the last thing I'll say is that the reaction is energetically favorable or thermodynamically favorable. So thermodynamically favorable. So what do I mean by thermodynamically favorable? I mean that the reaction is like it's going to proceed probably relatively easily. Okay, so if you think in terms of like equilibrium, it means that the reaction is favored to the right quite a bit. So what about on the flip side? What if the delta G is positive? So we can write a note about that. If delta G is positive, okay, so what if delta G is positive? Basically the exact opposite, okay? That means that the system requires energy, okay? So the reaction requires energy in order to proceed. See somebody has a question, I'll get to that in a moment. So energy, of course, usually means ATP, right? So I'll say that often in the form of ATP. And we're talking about usually anabolic reactions. All right, so I will show you some examples in a minute. Basically the same examples I showed you before in terms of glucose catabolism or building of proteins. But kind of this is the main thing to remember, right? If you see a delta G value, and you see this sometimes associated with certain reactions, to know what that means. If it's negative, it means one thing. If it's positive, it means another thing. Positive, like I said, it means it needs energy, or else it won't proceed. Negative means you can get energy from it if you break that thing down. So I don't know why that disappeared. What does this mean? It says energy is lost from living systems as entropy and heat. Only processes with a negative delta G are spontaneous, and they can be harnessed to do work. So back to talking about catabolic and anabolic, and notice this time I'm using different words. I'm using exergonic. So X means out, and gonic means energy, by the way. End means in, and gonic means energy. So it says here, exergonic reactions are things that lose energy, so the delta G is negative, or less than 0. An example I gave you before was the catabolism of glucose. And notice this has been measured. This can be measured experimentally, how much energy is released from the system. And notice it has a negative sign to that. On the flip side, we can also have endergonic reactions or anabolic reactions, and photosynthesis is a good example of this. And so notice this requires energy to proceed, in this case light energy because it's photosynthesis, and so our delta G has a positive value. And so that's kind of really what I want you to know, is what does it mean if you see the positive, and what does it mean if you see the negative? It's a pretty typical midterm 2 kind of question, is to ask this kind of thing, right? It might ask, you have a reaction and this is the delta G value, what does it mean? And then, I'll give you some options, and it means that if it's positive, it needs energy to proceed. If it's negative, it's going to release energy when the reaction occurs. So just a reminder, like I said, in terms of what we're looking at, catabolic pathways are basically exergonic, and anabolic pathways are endergonic. And so, these things are both important in our cells. Sometimes we have to break things down to harness energy. Sometimes we need to build things up if we want to, be organized, because of course cells do. We need to build things all the time. So these are just some graphs trying to show you what's going on. We're going to kind of come back to some of these graphs a little bit later on when we talk about enzymes. And it's just saying, hey, if you're an exergonic reaction, the reactants have a lot of energy, so complex chemical bonds, and they lose energy when they become products. And notice that energy is being released from the system. So that's a negative, going from high amount of energy to low. You can kind of think of it as the guy or girl, you know, jumping off of the diving board. On the other side, it's the exact opposite. Someone's climbing the ladder, or we're putting energy into a system. It's becoming more complex, and that requires energy. And so the delta G, of course, is positive. So like I said, remember what that means, positive and negative delta G's. That's kind of the main thing I want you to take away from this. So here's a sample question. Like I said, often I'll have, you know, usually I'll have at least one like this on the midterm. And so you can see it says here positive delta G of plus 2,700 kilojoules per mole means. And now you got to take a look and read all the questions and read all the answers. And of course, remember what all these words mean, right? The reaction will be exothermic. So what does that mean, right? Exothermic means releasing energy. So that's no, it's not exothermic. It's not releasing energy. Positive delta G means it requires energy to proceed. The reaction will require an enzyme. That doesn't seem to be relevant at all, so that's a no. Most reactions in cells require enzymes. The reaction will be spontaneous. I guess I didn't really talk about that. Maybe I should have said favorable in this, and that's a no. That would be negative delta G. The reaction requires energy to proceed. Yes, we already said that, right? Release that much energy, no. So this is the correct answer here. Requires energy to proceed. So make sure to read them all carefully. Okay, so moving on, let's talk a little bit about these cells and their energy and what's going on in these cells, right, in terms of how that energy is being used. So a cell is not at equilibrium. I know if you've taken chemistry classes, you know, chemists love to talk about equilibrium because you know what, that's what happens in test tubes. But that doesn't happen in cells. Cells are rarely at equilibrium. Cells are always consuming things and expelling waste products, and they're always building and moving and doing all sorts of fancy things. So this is kind of the model of what's going on, right? You can see nutrients and energy are going in, waste products and heat are being lost. It's more complex than that, obviously. So I'll come back to that model in a minute. You can see this is a bit more like what's going on in a cell. If this was at equilibrium, you could see this water level would eventually kind of level off and then it would stop. And cells don't. We're like multi-step reactions, but multiply that by thousands, because there are thousands of reactions happening, and we have these machines that have to run, and they have to run constantly. We don't turn them off, so we don't reach equilibrium. So here's our cell, right? And think about the possible energy sources we could have. As humans, we eat food, and most of that food is carbohydrates or fats or proteins. We do eat nucleotides, but it's not really a significant part of our nutrition in terms of getting energy out of them, right? It's just such a small part of the cells that you eat. If you're a plant, you might take in light energy. If you're a specialized bacterium, you might use certain types of minerals. You can see some bacteria use some weird things. So energy goes in, and usually what happens is as the energy is going in, as those nutrients are broken down, we convert that energy into ATP. So we'll talk about ATP in a minute. And then that ATP is used to build bonds in new molecules. So those new molecules might include all the usual suspects, our carbs, our fats, our proteins, and nucleic acids. And during this process, we're going to release waste. And so some of the energy will be released as heat, and some of the energy will be released in bonds in those waste products. So that's kind of a little bit more of a complex picture. So we do want to talk a little bit about ATP, and we want to talk about something that is called cellular work. So we'll get to that in a moment. So there it is. There's ATP. You know, nice molecule. If you don't know what ATP stands for, it stands for adenosine. So this here is adenosine. So adenosine. So that's the A. And then we have triphosphate. So that's T and P. So I think we talked about this molecule before, way back in topic 2. We were talking about macromolecules, and I think I had said, hey, what kind of molecule is this? And I'm sure everybody now, because you've studied for the first midterm, you're looking at this saying, oh, it's a nucleotide. Take a look at this. We've got a nitrogenous base here, we've got a pentose sugar, and we've got some phosphate groups. So it's obviously a nucleotide. So remember, I think I'd mentioned before that nucleotides are not just involved in information, like DNA and RNA, but some nucleotides are involved in energy and energy movement around the cells. The other thing I pointed out way back in topic 2 is that when I look at this molecule as a biochemist, holy cow, I see all these oxygens. Look at all these oxygens. Some of them have charges, lots and lots of charges. So what we have here is basically something that has come together, because we have all these negative charges, and you probably know from chemistry that negative and negative, they don't want to be together. We want a negative and a positive. So in order to get all of those things together, it would have taken energy, and that means that there's energy stored in these bonds. So this bond here and this bond here, there's actually a significant amount of energy stored. So ATP is a place where we can store energy, at least temporarily. It's not the type of molecule you want to store in loads because it's not as easy to store as other compounds and not as stable, but it's a good transient energy source. And so we use ATP to drive a bunch of other reactions, and the way we do it is we do the reverse reaction. We hydrolyze it. So this is showing a little stick figure of ATP. Let me just go back for a second. So by the way, each of these is a phosphate, we call it. And a phosphate is often represented with like a P with a circle around it. So that's what you're seeing on the next slide. So you can see we've got 3 P's with circles around it. Those are the phosphates. We've got the pentose sugar and the nitrogenous base. And when we break that down using a hydrolysis reaction, that releases energy. And so this is the whole point, this whole exercise is to store and release energy when and where we want it as a cell. So notice this P has a little I beside it. I'm not really sure why that's necessary, but often it's called inorganic phosphate. And inorganic means it has no carbon associated with it, but you can still see it's the P with a little circle around it. And so same symbol. So that just means there's a phosphate being released. So we're going to see a lot of reactions that involve ATP, by the way, over the next few units. So what is ATP used for? ATP is used for something that I called cellular work. So what is cellular work? Well, some of it is probably really obvious, moving things around. So you can see there's 2 examples there. We're using a little bit of ATP here, and it's being used on the top to pump some ions across a membrane. So we're moving things, and that's very obvious as work. You can see in the end we've got ADP. So D is for di, it's lost a phosphate, so it has 2 phosphates instead of 3, and it's got that inorganic phosphate there. You can see on the bottom we're moving something around as well, and in this case we're moving around an organelle, and that's more cellular work, right? So these things are obvious, and if you're an animal, maybe you move your muscles, and this takes a lot of ATP too. But that being said, a lot of ATP is used in something called energy coupling. So you can see there's that word coupling. And so what does that mean? Okay, that means that we're taking reactions and using them together to accomplish something. So here's our relationship between our exergonic and our endogonic reactions. You can see we're consuming food there on the left. So our food is in the form of some sort of macromolecule. We're going to use ADP and inorganic phosphate. And then we make ATP. There it is. Yay. This is not a long-lived molecule because usually right away it's used for something, often anabolic processes, and anabolic processes are going to basically mean we're building new macromolecules, or sometimes we're using it to do cellular work. And of course, in the end, the ATP is recycled into ADP and inorganic phosphate. So we talked about the cellular work here, and like I said, a big part of that cellular work is actually making new macromolecules. And this is in a process called energy coupling. So there's the kind of the definition of energy coupling there, right? It says ATP is passing off phosphate groups, it energizes things, And as things get energized, they are able to proceed along a metabolic pathway. So what I want to do is show you an example of energy coupling, and I'm going to draw something that looks a little bit like figure 8-9 in your textbook. So you can grab a piece of paper, and I'm just going to flip over to the whiteboard here in a moment and draw you some chemical structures and try to explain how energy coupling works. So let me just flip over to that whiteboard. There it is. Okay, I guess I got to change zoom to go to the whiteboard, and there it is. Okay, so I'm going to draw some things for you, and I'm going to show you what energy coupling is, looking at this reaction, which is the synthesis of glutamine. So glutamine is an amino acid, by the way. So maybe I'll put that in brackets. So an amino acid, an amino acid. Okay, so let's draw, let's show the reaction. I'll show you what, how this reaction works, okay? So basically we're going to start off with something called glutamate or glutamic acid. So it looks something like this. And if you don't remember all of your chemistry, that's okay. I like chemistry and I like chemical structures, so I like to draw them. I won't ask you to draw this one on a midterm or anything like that, but I'm just trying to show you what's going on for those, particularly those that do like chemical structures and like to understand what's going on. So that is glutamic acid right there. Okay, hopefully that's neat enough for you. So glutamic acid. So glutamic acid has an abbreviation, by the way. All these amino acids do, and it's GLU. Okay, so if we want to make glutamine, we are going to add this to ammonia. So ammonia is a simple compound. It looks like this. It's NH3. There we go. Okay, and there's my chemical arrow or reaction arrow. And so I apologize, I got to draw a bunch of this all over again. So here we go. All right, I'll show you what is going on with this. We have one more C and H. And then down at the bottom, almost all the same, but we've got to add that nitrogen in somewhere. And that nitrogen is going to get added down there at the bottom. So there it is. I guess I could do that in a different color for you if you like. Here we go. And of course, it's not entirely balanced, so I got to add water. Okay, so notice we're missing 2 H's and an O. Okay, I'm just going to shrink this just a little bit. I got one more piece of information to add to this chemical reaction, and that of course is our thermodynamics. So it turns out that the delta G on this reaction is plus 3.4 kilocalories per mole. So now you got to go back to your notes and you have to remember, wait a second, what does that mean? So this has a plus value, and that means that the reaction is not favorable. And so that means it needs energy to proceed. So needs energy to proceed. Okay, so, and you know, just going back to your notes again, you've got to remember, where do we get energy from? Well, we get energy, or we use energy in ATP. So you may have noticed, I'm just going to slide this up here, and maybe I will make it a little bit smaller so I can squeeze everything in here. How do I, there we go, right, squeeze that up to the top. So notice I put a 1 up there. I'm going to give you another chemical reaction. So chemical reaction number 2, and I'm not going to draw the structure for ATP. That is a big structure. It takes me a little bit of time. So ATP. And we can hydrolyze ATP. So hydrolysis reactions require water. And we would get ADP, and we would get inorganic phosphate. And so the delta G for that reaction, I have the number as well. The delta G for that is negative 7.3 kilocalories per mole. Okay, so notice with this reaction, maybe I will just, let's see here, I'll grab another color. So notice that the waters cancel out, and what you can do, this is how you couple two reactions. You kind of put them together, and the waters cancel out, meaning that there's water on both the reactant side and the product side. And so we can look at this in the overall reaction. So overall, all right, is we take glutamic acid plus ATP plus ammonia. Glutamine, by the way, I guess I didn't label that for you. I will label that for you right now. This is glutamine. And the abbreviation for glutamine, by the way, is GLN. So sorry for cramming it in there. Okay, make that a little bit smaller. Okay, so glutamine, GLN plus ATP plus inorganic phosphate. So, and you notice I basically just added everything together. And so you can do the same thing with the delta G. So I will do that. I will add the two delta G's together. So negative 7.3 plus 3.4 is negative 3.9 kilocalories per mole. So I just got to shrink that one more time. I guess I'm getting bigger and bigger with my writing, but hopefully you can see all that. So what we can say about this reaction is that therefore the overall reaction is favorable, meaning it can proceed thermodynamically and it has enough energy to do so. So that's what energy coupling is all about. And that is where actually most of our ATP goes into. So when I say cellular work, if I were to ask you, I think I did on the fall midterm, I asked, give 2 examples of cellular work, right? Or something like that. This is one of your answers, energy coupling. Another answer could be muscle movement or membrane transport or something like that. But this is actually the big part of energy coupling, or sorry, a big part of cellular work is energy coupling. Okay, so hopefully you got all that. As usual, if somebody would like me to share with them my diagrams, then, oh yes, I made a mistake. Somebody just corrected me. That should be ADP. Let's see here, I better erase that. There we go. Thank you for the correction. ADP. Somebody can add better than I can. So thank you very much, Michelle. Okay, so like I said, if you would like me to share this with you, just send me an email. I can always forward it to you if you didn't get all that. I can probably be a lot neater if I was on paper rather than the virtual whiteboard, but I think we did okay. All right, so back to the PowerPoint. Where did it go? I have to find it here on Zoom sharing. I know it's here somewhere, I just don't, oh, there it is. Okay. So like I said, this is energy coupling. And you can see what the textbook does. They do it a little bit different than I did, but it's kind of the same idea in figure 8-9. That's my reminder to do it. I was going to do it on this PowerPoint, but I figured there's more room if I did it on the whiteboard. So that's actually it for topic 8. And topic 9 is on enzymes, and I'll finish a few minutes early today. There's no point in me starting topic 9, but I'll just say a couple of things about enzymes is that if metabolism is all of these reactions going on, there's thousands of them, and that's what's shown in that map there on the right. This little pathway that you're seeing on the right, so starting at this point here, this is actually glycolysis, And then we've got the citric acid cycle there. You may know the citric acid cycle as the Krebs cycle. And that's just one set of reactions in the cell. There's hundreds, thousands of reactions going on in cells. And almost all these reactions are catalyzed by enzymes. And so it's important that we talk a little bit about enzymes and what they are and how they work. And that will be the next topic. So like I said, we only have 5 minutes left. There's no point in really starting the next topic. So it's Friday afternoon. It's a good day to get off a few minutes early. So I hope you enjoy your weekend, and I will I'll be looking at your reports a little bit this weekend, and I'm not going to comment on whether that's enjoyable or not, but hopefully I'll be able to get those back to you soon. So have a good one, and I'll see you next week.